Okay, thank you, and thank you for the invitation to present this. So I'm going to present some work on exosomes that we are doing in collaboration with Carlos Cordon-Cardó and Ash Tewari. So exosomes are relatively old. They were christened in 1980 after being observed in electron microscope images. And then some of its dynamics was described in 1985. So the idea was that basically there are two invaginations of the cell membrane. One produces an early endosome, and that early endosome invaginates again and creates exosomes inside that take some of the cytoplasm of the cell of origin. And then those are, the multivesicular bodies are fused with the membrane, and then the exosomes are released. And this, when they are released to the body fluids. And exosomes have been found in all body fluids, including cerebrospinal fluid, urine, blood, and so on. So because of that, there was very much interest in it. Where the rate of publication has increased such that more than 80% of what has been published in the last few years. And so what has been described is exosomes have microRNAs and RNAs inside, which is a good discovery in 2007. And since then, there were applications in many areas, including urogenital cancers. And one of very interesting recent papers by Michael Donovan and others is that there can be a prediction with an area under the curve of about 0.87 to describe Gleason 6 or less, or Gleason above 7 from exosomes. So the problem with exosomes is that they are very small, 100 nanometers, and they are very difficult to isolate. And so the idea is that most of the people isolated with ultracentrifugation or filtrations of different sorts. But the thing is, there is a lack of reproducibility. Different labs try to find it, and they don't find the same thing. So what we did was try to imagine what would be a good filtration system that would overcome some of these difficulties. And what we developed in my group is a system in which basically you have a pillar array with some asymmetry that allows you to separate small particles. They would go straight, and big particles, they would bump against these pillar arrays. We described this in a recent paper. So the idea that what I'm going to show has been looked at under a microscope, and this is the array, and inside the array there is this pillar system, and this pillar system basically is 1,000 times smaller than a hair. So what happens is that basically you have here a 100 nanometer particle and 50 nanometer particle, and the 100 nanometer particle, which is typically the exosome size, goes to a side, and you can collect it. And you can do it in such a way that is very friendly. And so basically this image, which I cannot show, no. Would show that basically you put your chip in a cartridge, and it starts to do the job of isolating exosome from a patient sample, for example, in about 1 hour. So by isolating exosomes using this technique, you can separate some prostate markers. In this case, it was GATA2 and PSMA, and you can separate cancer from non-cancer with an area under the curve of about 0.89. And right now we are trying to analyze it using RNA-seq. And interestingly, what we have found is that there is a very clear signature of the cell of origin, which is these pileups that disappear in exosomes, but also in the cell of origin in a sea of nothingness. So you can try to isolate what comes from where, because exosomes are coming from all tissues, right? And the idea is to try to figure out which is the tissue of origin. And if you identify that, for example, it's for prostate cancer, then you can try to figure out whether, from a large cohort, whether you can distinguish indolent from aggressive cancer, or who is going to go into biochemical recurrence. And so this is work that we have done with my team, some of which work directly in Carlos's lab, and with their guidance in the pathology and the samples from Ash Tewari in the prostate cancer patient samples. Thank you.